right. Hello, students. Welcome back to I believe, sixth week. Sorry, I'm losing count of Biology 1406, Intro to Biology for Science Majors. This week, we are going to be talking about metabolism. Let's check to see if my pen is working. It is working just fine. So, it's a word you've probably heard before, but what is metabolism? Well, a way to think of it is metabolism is just energy, or it's a way to conceptualize energy because energy flows through all living systems. Um, it is what powers our chemical reactions. It's how we sustain life. Anything that has motion, um, that moves, that breathes, it needs to use energy. So it's what's happening inside every cell. And metabolism just basically transforms that energy into action. And that's why metabolism and energy are just inseparable. You cannot talk about one without talking about the other. Okay? So metabolism is the sum of all chemical reactions that sustain life. So if you are living, you are metabolizing. Okay? Life is metabolizing. It is a series of metabolic reactions. And how this happens, how we get to this point that we can sustain life, is we have to use energy. Think of it like gas in a car. You cannot drive your car without gas. It needs energy. But what is energy? It's a very important question. And if you're not familiar with it, um, you may just overlook it without completely understanding what we're working with here. So today we're going to talk about a few things. We're going to talk about, obviously energy and metabolism. We're going to talk about the different energy types, specifically potential and kinetic. We will get into free energy in the form of Gibbs free energy and activation energy as it pertains to enzymes. We are going to talk about the thermodynamics laws. Now, you might be asking, what? Why that? Well, remember you had to talk about chemistry to understand biology? Well, unfortunately, you can't escape physics either. Okay? That is very important. We're also going to get our formal introduction to ATP, which is the abbreviation for adenosine triphosphate. Okay? And how do we get energy to here? Okay? So that's what we're talking about today. So, um, where do we get energy from? Well, we get energy from, and the short, answer is we get our energy from our food. So food equals energy. Now the question is, how does food have energy? Right? So again, to trace the conceptualization back, how do we get energy? Well, cells, we need nutrients. We need proteins, we need carbohydrates, but what is energy? It's important that you understand this because with metabolism being the sum of all those chemical reactions, we literally have two things that we can do in a metabolic system. Okay, the first thing we can do is we can have anabolic pathways. Now, anabolic pathways, we can build stuff. Catabolic pathways, we can break stuff down. These are the two things that can happen in a body or in a system, um, and both deal with energy. With anabolic pathways, and a way to look at this anabolic build, think of anabolic steroids. If you've ever heard that term, that's the bad stuff. That's the Barry Bonds stuff. Um, they build muscle. Okay. Now, if I'm building something, I require energy to do that. Okay. I've put energy into the system to build it. Now, a catabolic pathway, which breaks stuff down, a catabolic pathway will release energy. Okay. So an anabolic pathway builds. An anabolic pathway, um, an example of anabolic pathway is something like protein synthesis. Like we talked about this when we talked about macromolecules. So protein synthesis Or I take amino acids and I build them into protein. Okay? Through those peptide bonds. Remember? A catabolic pathway, it would just simply be the reverse. It's if I were to take a protein and go to an amino acid. You could call this digestion, right? So when I break stuff down. So the idea behind metabolism is I can spend energy and then to make something. So, or I can, but I'm going to do it a better way. I can deposit energy into the bank, and then I can withdraw energy when I need it. Okay? So I can build stuff up, I can deposit it, and then I can withdraw it should I need it. So it's kind of like this economy, so to speak. So think of it like an economy, and I'll just write economy here up at the top here. Okay? Where I'm depositing and withdrawing constantly. Now, I don't ever want to deposit too much. I don't want to withdraw too much. I want to exist in this equilibrium. Um, Life really depends on the balance between these two things happening and not doing it too quickly. And we'll explain why. So what are we doing here? Let's go to our first Blackboard slide here. So what is energy? Well, what is energy? And on some level, where does it come from? Right? That's where we were really, because again, will I quiz you on this? Probably not. Will it help you understand what we're talking about? Absolutely. So, um, I may quiz you on it. I don't know yet. But we talked about a few things uh, already. So, what is energy? Where does it come from? Okay. Let me get another color here. So, we've talked about energy in the form of glucose, C6H12O6, right? And that we can get energy from breaking these bonds. But how do we get that energy? It's not just this material thing that transpires. If we trace it back, how do we get glucose? Now we'll talk about these steps in kind of reverse, but we'll talk about these other steps in a couple weeks where you have glucose. Well, how do you get glucose? Well, plants make glucose in a process called photosynthesis, okay? Where you take light and you make glucose, right? You may not know this. You don't have to know this just right now. But we as humans, we will, if you want to trace like this food chain back down, essentially what you're doing is, let's just say you're eating broccoli or a leaf, whatever. That leaf has glucose in it, but how did that leaf get the glucose? Well, it made the glucose. It makes glucose in a wonderful process called photosynthesis. So, but how does that process start? Well, it needs this light. And where does it get this light from? Well, it gets it from, you guessed it, the sun. All right. And that's the key concept that I want to talk about right now is all energy is from the sun. Okay. So energy comes from the sun. So where does sunlight get its energy? So for that answer, you're going to need a brief trip into physics. Um, so let's talk about the sun here. So what is it? What is the sun? Well, it's not just the big yellow ball in the sky, as a certain comedian said. So what is the sun? Well, you have to think of the sun as this big fusion reactor. Come on. Fusion reactor. All right. And to understand what this means, fusion reactor, I'm going to take a step back because you've probably heard of this guy called Einstein, and he had this equation that said E equals mc squared, where he talked about energy equals mass times, and I'll just say speed of light. That's speed of light squared. So energy equals mass times the speed of light squared. And he postulated that energy and mass are linked. So why is this important? Just a little bit of mass, and again, if I multiply it by the speed of light squared, speed of light is very big, just a little mass can give me a lot of energy. And I will show you what that happens. Back to our fusion reactor, and specifically a hydrogen fusion reactor. Okay? So when I'm looking at this, when I'm looking at the sun just has a bunch of hydrogen floating around in it. Not really floating around, but it's just basically taking hydrogen, and I will separate this right here. And remember, hydrogen loves to exist by itself, or loves to exist in concert with another hydrogen. That's why you have H2. Now, in a fusion reactor, we're fusing hydrogens together. We're fusing hydrogen atoms together at a very explosive rate. All right? Hydrogens are fusing together. And when I create those hydrogens and I fuse them together, I make helium. All right? Because remember our atomic number. If I can just fuse two protons together, they basically make helium. If I get two protons into a nucleus, that's helium. One is hydrogen, two is helium. Okay? Now, if I take the atomic mass of hydrogen, hydrogen has an atomic mass of 1.0078. Helium has an atomic mass of 4.0026, or 4.0026, 0026, okay? 0026. Okay? Now, where am I going with this? Well, in this case right here, I have four hydrogens fusing together. So really, if I have H4, I end up with an atomic mass of those four elements hitting each other of 4.00. Sorry, that's not a zero. I'm going to erase it. 4.0312. And helium is still at 4.0026. Now, there's some neutrinos, there's some positrons going on here that you don't need to know about. But what's the idea? What I'm getting is I'm taking four hydrogen, fusing them together to make a helium, and what I end up getting is a delta mass, a difference in the mass of 0.0286. So helium is 0.0286 atomic mass units lower than those four constituent parts. So where did the mass go? And the answer is it became energy. Because if we go back to Einstein's theory, energy and mass are linked. So just that small difference in that mass, fusing it together produces energy. And that's what our sun is doing. It's fusing hydrogen together all the time. But it's doing so in a very interesting way because it's making heat, it's making light. Go back to our slide here. Keep going. Where's our slide? Did I not do it yet? Okay. So if I go back to my slide, that's where energy comes from. I think I did it on the whatever page I did it on. We won't worry about it. But again, the sun makes energy through fusion of mass. Okay? Hydrogen becomes helium, we have a release of energy, okay? A fusion bomb, a fusion reactor. So matter itself contains energy. This is happening in the sun. Mass becomes energy. So I need you to remember that. So the energy leaves the sun as light, heat, electromagnetic radiation, and it travels to the earth, and that's what fuels life. So if I were again to map this out, we do this as a little map here. You have the sun, and through fusion makes energy. We'll call it light. Okay. Plants, through a process of photosynthesis, take that light and they make glucose. Okay. And then I sum this all the way down here. So we have consumers then, which we'll just call ourselves a consumer. We are consumers, we're humans, but so are antelopes or deer. You get the idea. Take that glucose to make ATP, okay, from ADP, okay? And what you have here is you have two components here. Let me get my ink here. So up here, the sun, all this stuff, you have photosynthesis. And right here, this stage, that second stage, you have respiration. You have human respiration. All right. So photosynthesis, respiration. If I look at that from a mechanical or a, how do I want to say this? I look at that from a formulaic standpoint. Well, this is what it looks like. Let me get another color here so I'm not confusing anyone. I'd hate to do that. Is if I'm looking at photosynthesis and I look at plants. Well, I'll look at I'll look at plants first. Um, and plants, they take some C. That's you can't read that. Why am I doing that? That's terrible, guys. Okay, we'll just take the basic formula here. We'll do some CO2. So remember, plants breathe in CO2. Um, they take in. That's also bad too. I'm fumbling today, guys. This is not good. They take in some water. Okay. And what they end up yielding here, and I'm skipping a step because I forgot about the energy. Okay. They take that light energy from the sun, and what they end up yielding is C6H12O6 plus some oxygen. All right. Now, I haven't balanced this equation. If you really want to get technical, it's 6, 6, and 6. And if you were to count all the numbers there at that point, that's what ends up happening. You get that balanced out. But 6 CO2, 6 moles of CO2, 6 moles of water plus light energy from the sun is going to yield glucose and oxygen. Plants breathe out oxygen. Us, on the other hand, we take in O2 along with some glucose. We eat the glucose, and what ends up coming out of that is CO2 plus H2O plus energy. And that's where we get the energy from, okay? It's harvested in this process. And again, if you want to be precise about it, that's the precise nature um, of what's happening here. So can you follow the energy? Fusion converts a tiny amount of mass into energy. Sunlight reaches the earth. Plants capture that energy. Energy becomes stored in glucose. Okay. That's photosynthesis. Animals consume the glucose. Um, they break down glucose. We produce ATP. That energy is all transformed or transferred from each other. Okay. So what are we talking about when we talk about energy? Well, the big ones that we talk about here, when we talk about energy is, and I'm still on my white marker here, so let's get some dark blue here. We'd have some kinetic energy and we have potential energy. There's other types of energy, but the two big ones we talk about are kinetic energy. Chemical energy we talk about when we talk about ATP. Um, Heat, anything with movement is produces energy or heat. The sun produces light energy or electromagnetic radiation, right? And then electrical energy in the form of electrons, like a battery. Okay, those are all different types of of energy, but the two overarching types we talk about is energy of motion and energy that is stored. So an energy of motion is if people are walking, if people are a river is flowing, so like this hydroelectric process, that's kinetic energy. It's a moving, molecules vibrating. That's how your microwave works to create friction, to create that thermal heat energy. Um, this is kinetic energy. Now, the opposite of that is going to be potential energy. And kinetic energy, it's energy in motion. Then you have potential energy, and that's just energy that is stored. And the classic example we give is a spring. So this is my terrible spring. If I compress it, if I manually compress it and make this tightly wound spring right here, well, this is stored energy here, okay? As I'm inputting energy into the system here. And this spring is trying to push back, but I've compressed it to the point where it doesn't want, or it can't go until I release it. Once I release it, it then becomes kinetic energy. Okay? So this is stored energy. Um, Another example could be, you're at a hill, there's my terrible hill, and you have a ball at the top of the hill. So the energy it wants to go down here, it has once it starts rolling, yeah, it has energy because gravity's pulling on it, but there's something stopping it. There's energy forward pushing up at the top of it. There's an opposite reaction that's happening to keep it motionless. But at the top of the hill, once I push it, it becomes kinetic energy. Okay? So it's important to in biology, the most important form of chemical energy is stored within molecular bonds. So when we talk about molecular bonds, if I can go back to this guy right over here, so something like glucose, when I had, that's too dark. When I had, think of my red here. When I had C6H12O6, you have chemical bonds. And chemical bonds are what hold that energy. That's where keeping things together. So without it, you wouldn't have these molecules bound together. It's pretty cool. So let's go back and let's get into a little bit of physics here. You may not like this, but it's important. It's important that you understand this. So we have a few laws of thermodynamics. I'm going to focus on the first two. And the first law of thermodynamics is that energy cannot be created or destroyed, only transferred or transformed. And energy equals mass. Don't forget that. E equals mc squared. Your book doesn't talk about this, but the conceptualization of energy and mass being the same thing, mass in the universe and the energy in the universe is constant. It will never go up, it will never go down. It's always been there. Creates a bit of a mind meld for you, a mind trip for you, but energy and mass cannot be destroyed. So if I use mass, remember I had my example about losing weight? You're just breathing out atoms, you're breathing out mass, you're using energy. So energy is not created, it's destroyed, only transferred or transformed. Go back to this guy right here. The sun, the energy, its mass, all that mass gets transferred across this entire process through photosynthesis, respiration into energy into your chemical bonds. Okay? So, going back to the laws of thermodynamics, you cannot create, you cannot destroy energy. Just you can transfer or transform it. Remember that. And the second law The second law basically says, and this definition doesn't really help. Um, the way I summate it is entropy always increases. And what is entropy? Entropy is just disorder, or a measure of disorder is disorder. That's terrible, but you can see it, I think. Entropy is disorder. It's a measure of disorder or randomness. So no energy transfer is perfect. Let's just say I go from one state to another state. I'm going to lose some of it as heat. Um, but the system itself, there's always tends to want to be in disorder. And where you see order, you're going to see energy input. Us as living things, we're highly ordered. We have organ systems, our cells have organelles. It is a constant battle against entropy, which again is disorder. So, um, we have to constantly consume energy to maintain that order. But the universe's total entropy is always increasing. So we're always moving towards disorder. There's signs of order being put together, but really we're breaking down faster than the universe is. Um, a good way to look at this is without energy, if you don't eat your food, remember food has energy, um, you're going to lose your proteins are going to degrade, your cells are going to deteriorate, organization is going to be lost. Think about something that passes away, a post-mortem individual. They begin to deteriorate, they begin to break down. No more energy is being put into the system. So if I go back, so if I talk about we'll just do energy again. Eek. We'll just do energy and laws. Okay. So we have the first law that just basically states energy is conserved, or yeah, energy is conserved. And the second one that just basically states we move to randomness. Right? That just tells us how nature kind of operates. Let me give you an example to kind of to kind of illustrate this a little bit better. So, in the case of, say, something like ice, here's our ice cube right here. Okay, ice is just basically water molecules, okay? And when they're frozen, they're tightly compacted, they're highly ordered. But eventually it'll move into a puddle. And again, you have a little bit more disorder happening right here. Basically, what you have is you have a system here with low entropy in the ice. There's low disorder, there's highly ordered, and you have high entropy over here. Okay? Low entropy to high entropy. And the low entropy um, is just basically saying, okay, I have a lot of order here, but the system tends to want to move to disorder. So our universe tries to push these things into a certain level of disorder. The system wants to move in this direction, okay? More disorder, okay? And that's what entropy is doing. But we don't lose any mass there. We're not losing anything. We're just moving to randomness. That's what water does. Now, this doesn't tell the whole story because then, what's even more random is steam, and that doesn't necessarily happen randomly, right? That's even that vapor that's coming up, that's not happening naturally. So what's going on here? Well, there's more to the story because neither that just tells us what the system wants to do. Um, but nothing tells us whether it will happen. Okay? So we'll leave the ice example right here because I'm going to talk about Gibbs free energy. Okay? Gibbs free energy. Cells need to be able to use energy. Um, and this energy, we define it by this term called Gibbs free energy. Okay, so Gibbs free energy, it's just named after the guy. Um, is named after the guy who did this. I forgot his name, but his last name is definitely Gibbs. It's in your book, I'm sure. I don't care if you know that. Okay. And so Gibbs free energy is marked by this formula, and I'll break down this formula. It's delta G is equal to delta H minus T delta S. Okay? What does this break down to? Okay? Let's change my colors here. Okay, so delta G is Gibbs. This free energy, Gibbs free energy. Delta H is a measure of enthalpy, which is the amount of energy in the system. Or the energy that can be released, so to speak. Um, Delta S is a measure of entropy. Remember, that's disorder, how disordered the system's going to be. And T equals temperature in Kelvin. And if you're unfamiliar with Kelvin, that's just Celsius minus 273. Okay? Where a temperature of 0 Kelvin is absolute zero, where no metabolic processes can happen. That's actually the third law of thermodynamics. Absolute zero. Okay? So delta G, Gibbs free energy, delta H, enthalpy, the amount of energy in a system. Okay? And entropy um, is what measures disorder. Okay? So if I go back to our ice example, and remember, temperature is a factor here. Okay? So if temperature isn't high enough, it will have a factor. So I get temperature high enough, yes, this water will move to steam. So temperature has a factor in this that we don't see. So hopefully that helps clear some of that up. I know it doesn't clear all of it up just yet. So Gibbs free energy, and those are the breakdowns of that. And I will get my white pen here. Okay. I, I'm not going to ask you to calculate anything. Okay. Just know this formula because the big thing that you're going to want to know is if delta G is less than zero, if delta G is less than zero, um, we call this exergonic, which means it will happen spontaneously. And if delta G is greater than zero, we call this endergonic. Endergonic, G-O-N-I-C. Okay? So exergonic, spontaneous, spontaneous. And endergonic means it needs energy to happen. It needs an input of energy. So in some cases, that could be temperature, that could be light, but it needs energy. If delta G is less than zero, and again, if I go back to this, again, I have an entropy measure. I'm increasing my entropy. I have a temperature, and then I also have a sense of energy that's going into this system or being removed from this system. At this particular temperature, my delta G is less than zero. Now, at that same temperature, this delta G is greater than zero because it's not happening. It's going to need an input of energy for it to happen. Okay. Now, if I increase the temperature, this delta G could flip. Okay. But it doesn't in this particular case. So know that enthalpy is the energy in a system, um, that entropy is a measure of it, and that temperature has a factor in it. The higher the temperature, the higher order of randomness happening. If I increase the temperature so high and increase the ability to move to randomness so high, it will the system will eventually be an exergonic reaction. The higher the temperature, the higher the disorder, the more prone to spontaneity it's going to be. Okay? So, if I could just break these terms down a little bit. So, exergonic, what does this look like? Exergonic is going to release usable energy. Usable. Can never remember how to spell usable. Okay? It's going to release usable energy and it's spontaneous. All right? It just happens. Um, we have something like ATP hydrolysis, where we make or we release where ATP bonds are broken to release energy. Respiration. Okay? All this is exergonic. It happens. Another thing in a non-biological manner is combustion. Okay? And then we have endergonic reactions. I hope don't run out of space here, which require energy and they're not spontaneous. So I'll just put not spontaneous. An example of these are anything you synthesize stuff with. So remember we talked about this, like protein synthesis. These don't happen naturally. It needs an input of energy. DNA synthesis, photosynthesis, you need energy for these things to happen. So if I were to ask you on your exam, is the delta G of photosynthesis greater or lower than zero? Well, it's greater than zero because it's endergonic. It needs energy to happen. Is the delta G of ATP hydrolysis endergonic or exergonic? Well, it's exergonic because I am it's spontaneously happening. It doesn't need energy for it to continue. And another thing to happen is as I break stuff down, remember, breaking down stuff, breaking down stuff increases entropy. So as I break stuff down, entropy increases. As I build stuff up, entropy decreases. So think of it in that term. As I start to build stuff up, again, entropy goes down. As I break stuff apart, entropy goes up. So in that particular case, if I start to build too much stuff up, the temperature is not high enough. Doesn't matter how much energy is in the system, I'm not, or how little energy is in the system, the delta G is going to be greater than zero and it won't produce without any energy input. And again, that's completely dependent upon the temperature, atmospheric pressure. But the big thing I want you to look at um, is endergonic versus exergonic. Now, I want to dovetail into this because I don't want you to get confused. You may not have heard of these terms, but similar terms that you may have heard of um, are so we have endergonic and exergonic. I do not want you to get these confused with endothermic and exothermic. Okay? Okay. Endergonic and exergonic have to do with that delta G, that Gibbs free energy, and whether or not a chemical reaction will occur. Endothermic and exothermic have to do with that delta H, the enthalpy of the system. Okay? Endothermic absorbs heat or removes heat from a system, so it gets colder in the environment. Exothermic releases heat. Okay? So like your heat packs and your ice packs are good examples of that. But those deal ectothermic and end or yeah, ectothermic and endothermic or exothermic and endothermic deal with this delta H. Okay? Whereas endergonic and exergonic deal with delta G. So to put this into an example, you can have something that's endergonic and exothermic. You can have some, these can combine with each other. All right. But the example of something like that, say that's endothermic, And again, I just don't want you to get confused because I used to get confused about this. So something that's endothermic is like melting ice. Like the ice is pulling heat from the system. It's pulling the heat from the room. Like if you were to measure the temperature around the ice, it's colder than the room. All right. Melting ice, like sweating, would be an example, right? You get cooler whenever you sweat. Exothermic reactions release heat. This is something like fire. Okay. Steam condensing, and combustion reactions. So combustion, right? So don't get those mixed up. I really want to make sure you understand this. I'm going to ask endergonic, exergonic, because a system can get colder, but it's still exergonic because it's happening spontaneously. It's a much different measure of energy. We're measuring energy, not necessarily heat, but heat is important. So keep that in mind. So um, one other thing that I want to talk about um, is I want to talk about something, and this is just because it's in your it's going to be it's important to understand, but it because I'll break it down even further. But it's this idea. It's in your simulation today that you're going to be doing, or your for this week, is Hess's law. Okay, and Hess's law just basically states that the total energy of a reaction depends on the starting materials and the final products, not the pathway taken. I know that's a lot to work with, but we have energy is basically equal to total energy is equal to the starting materials minus the final product. Okay? So it doesn't matter the pathway that it took. So let me give you an example. Imagine you're driving from Huntsville to Houston. So Huntsville to Houston. All right. You can take um, you can take different routes. You can take 45. You can go around the Huntsville forest. You can go all over the place, but your starting points and your ending points remain the same. Hess's law kind of says this, where basically the routes may differ. I may take a bunch of different routes to get to the final product, but the overall energy that I've consumed or used doesn't change. So if I go from if I consume energy at step, or I start with energy at step A and blow it all at step B, or I go from A to B to C to D to E, which is the final product. So start, stop, start, stop. That doesn't matter. The amount of energy consumed doesn't matter on the intermediary steps. It's just the first and last product. Okay? So why does that matter in biology? Well, let me give you an example here. Okay? So Hess's law, we're going to separate this out, and I'm going to look at well, we've talked about this earlier, but you're not going to see this. I don't think you're going to see this anywhere else, to be honest with you, and I kind of like that. So we talked about respiration, and what's that formula? That formula is we take our glucose C6H12O6, we add a little bit of water, or a little bit of oxygen, sorry, and we get 6CO2 plus 6H2O and some energy, okay? And energy. I don't have room to write that there. You've seen that. But what about combustion? All right. What about a fire where I'm burning wood? So wood. Well, what is wood? Wood is just cellulose. What is cellulose? It's a long branch chain of C6H12O6. I add a little oxygen to that. I get CO2 plus H2O. And yes, one of the reactions of fire when you burn wood and you need oxygen. So if you've ever had to, you have to breathe, um, you have to blow onto a fire to give it oxygen. Well, that's why. It needs oxygen to start a fire. So wood burns and CO2 is produced, a little bit of water, which is vaporized as steam. Um, but that's the same process that's happening. You burning wood, if you were to eat wood, so to speak, which I guess you don't really want to do, but eat cellulose on some capacity, maybe like a plant, a cell wall, or something like that, or a cow does it. We'll just say that because they can digest the cell wall. They're going to burn that energy, get the same amount of energy as if they just threw it into a fire, because the starting points and the ending points are the same. Whereas in respiration, we're just doing it in a step by step by step process. We're doing it micro step at a time so we don't blow ourselves up. So the reactants are identical, the products are identical. The intermediary steps, just because it's doing it in a stepwise process, so combustion is just going, respiration is going tick, 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 tick. But the start points and the end points are the same. That's the difference. That's why Hess's law is important. Fire releases it all at once. Cells gradually do it to capture ATP. Because in this particular case, you get a bunch of heat loss, but you're still using the same amount of energy. Okay? So, taking all that into consideration here, um, is this the right one? Let's see. So yeah, enthalpy, I talked about that. I don't think that's what I wanted to talk about though. I want to ask you this question. How hot is steam? So, um, because you if you've ever held your hand over steam, like again, you don't want to get burned by boiling water. You don't want to get burned by steam. I asked this question to kind of bring this energy into focus and how this matters. So, um, if I were to like gun or took a thermal gun and do boiling water, so I say boiling water and steam, they're both going to be at 100 degrees Celsius. But why is it that steam burns so much more? Like if you were to put your hand in boiling water, don't do this. Please don't do this. It would burn. Yeah, it'd be bad. But it's so much worse if you get hit with that same amount of volume of steam. Well, why is that? Well, let's just take our stuff that we've been talking about here. So, let me go back to here. How did we convert? How do we convert? Um, Again, we had low entropy here. How do we convert um, this water right here into steam? Well, in this particular case, remember, it's not an exergonic reaction. It needs energy. Energy in the form of heat. Remember how I said if that temperature goes up, this will naturally happen? So basically what I'm putting into this system is energy. Okay? Even though it's more random, I'm putting energy into this system in the form of heat. Okay? So energy is kind of living in this steam. So coming back to here, where is my so in this particular thing is steam has energy that water does not necessarily have at that moment because again water doesn't have the energy that steam has. You have to put energy into the system to get the molecules to uh to be more random. Again, it doesn't exist at that way at room temperature. You need a lot of temperature to have that, which temperature is just heat, it's just energy. So in the system, steam has energy. Now, what ends up happening is if I cool that rapidly, the steam becomes water. But what happens to the energy? It's got to go somewhere, and it goes on your skin, and that releases energy, and that's what creates those steam burns that are just so bad, is that just release of energy. Steam is very powerful. It just releases so much. That's why you get steam engines, steam locomotion, is we just kind of capture that energy when water um, is superheated to the point where it just releases that steam. That steam condenses, and it just releases that energy, and you can power stuff. But if it hits on your skin, much more, a much worse burn, like 7 or 8 times worse. So 100 degrees Celsius, it would be like a 700 degrees Celsius water burn if water could get that hot. So steam has a higher entropy, like again, enthalpy increases, entropy increases, and both are represented in that Gibbs equation. Again, let's go back here. Let's go back to Gibbs. Again, when that heat energy goes up, when that entropy goes up, all that is energy. And then water has lesser energy, but the energy has to go somewhere, and it just goes on your skin. So the bigger lesson here is temperature alone does not determine the energy content. The state matters, enthalpy, entropy, all of that matters. So when you're looking at something like we talked about this fire is just combustion. It's releasing energy. So the question I have to ask you is once a system starts, so let's look at combustion. We talked about combustion just happening and that these are exergonic reactions. They happen naturally. They just happen in a state that you don't have to put energy into it. It just happens. So you're probably thinking like, okay, um, Chaz, how does fire, how is that exergonic? So fire or combustion, how is it exergonic? Or how is it spontaneous? That doesn't make sense because everything isn't just catching on fire, okay? So burning wood is thermodynamically favorable, yet why are our forests not on fire? So our forests are not on fire. These are very astute observations if you're asking this. So this is where we talk about, we're going to get back into the biology of things and just physics in general, is we're talking about fire and we're talking about why things don't just happen that just happen, or why things don't just combust naturally, spontaneously combust, is we're dealing with something called an activation energy. Okay? And just like that boulder on the hill, which again, once the boulder gets rolling, it's going to go down. But it requires a little bit of input. It requires a hill to come over, and that's probably not the best example. Here we go. Let me erase all that because the better example here is, let's say, here's my hill and the boulder's right here. Okay? This line right here, this line, this little hill right here is called the activation energy. Okay? Once I can get over that hump, the system will proceed as needed. Okay? Activation energy is key. Every reaction requires activation energy. It's like this built-in governor that prevents stuff from just happening and falling to pieces. So yes, it's spontaneous, but I need a little bit of a input of energy, or I need something to happen that allows me to proceed in this spontaneous manner. So it's required, this is what's required to initiate a reaction. As far as fire goes and lighting wood up, that activation energy, we could call it a spark, right? So I spark something up, I take a lighter, I light it up. That little spark produces enough heat to break the bond. Once I break that bond, as long as I keep producing wood or producing something that can burn and oxygen and the stuff it needs, it will not die, which is why the sun is kind of doing what it's doing up there. It's not dying. Once it's reached that activation energy, the system will continue to produce spontaneous or this it will produce exergonic reactions spontaneously. It's really hard to conceptualize, but activation energy is just kind of built into every system. Um, you need that to prevent stuff from just happening instantaneously. Yeah, so before bonds can break, and again, we were talking about wood, that cellulose, that C6H12O6. Before those bonds can break, you need an activation energy before combustion can happen. And if you think about your car, that's why you have spark plugs. It produces a spark to get the gas to break those hydrocarbon bonds to produce that energy to get your engine running. So yeah, activation energy is in every system. Without it, your ATP couldn't would instantly hydrolyze. You would just be burning energy all the time, and you'd have that heat build up. Your temperature would go through the roof. Um, DNA would degrade, biomolecules would fall apart. You have to have something that holds everything together. Um, and then within that, you need to have something that can overcome this activation energy. So yeah, the boulder analogy works really well. So this valley, th this boulder, this hill is kind of representing this activation energy. The ball wants to go down this hill, right? It actually wants to go from here to here, but it can't because it's got to overcome this. It's got to get over the hill. So the spark would be a match, spark, lightning, anything. Um, Once a few molecules react, the energy will just keep feeding itself. So what does that mean? It means that at the end of the day, we need stuff to harness as energy, but we also have this activation energy to make sure we're just not spending frivolously. Okay, it's sort of like this governor on a golf cart that prevents you from going too fast too quickly. All right. So what does this mean? Okay, so activation energy. Oops, didn't mean to do that. There we go. There we go. Activation energy. Let me catch my thoughts here. Um, what ends up happening here is we're able to store some of this energy. Again, we get energy from the sun. We're able to store some of it, and because I want to get different colors, we're able to store some of it in the form of ATP. So what is ATP? Well, ATP just stands for adenosine triphosphate. Adenosine triphosphate. Okay. So, oh man. So yeah, adenosine triphosphate, ATP is the powerhouse of the cell. Um, and there are three components to it, and I'll draw this out just because your book does it. Um, but essentially, you have three components. You have an adenine R group, you have a ribose sugar, and then you have your phosphate groups. Okay, you actually have three phosphate groups, but I'll draw this out right here. So again, you have your P. I'll just draw the phosphates like such. And then you have your ribose sugar here, which again, your ribose, come on, your ribose sugar is that 5 carbon sugar. And then you have your adenine up here. And I won't draw that out, but your ribose sugars right here. OH, OH. And you have your O group right up here. And then your phosphates are just bound to these oxygens, like so. And they're slightly negative in nature. Doom, 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 doom. Okay. So those are your phosphate groups, and I misdid this one. This one's double bonded. These are all double bonded up here. So get rid of that. All right. So that's essentially what adenosine triphosphate is. Now you have three phosphates here. You have your alpha, your beta, and your gamma. So really, when we do ATP hydrolysis, when we're breaking down ATP to make energy, ATP becomes ADP, ADP plus that phosphate group. It cleaves this gamma phosphate. So adenosine triphosphate becomes adenosine diphosphate. And that's how we harness that energy. So going up to ATP here. ATP. The formula we're looking at is ATP. And again, we're doing this form of hydrolysis. We're adding water. That's why I need to drink water, guys. Is going to yield ADP plus a phosphate, an inorganic phosphate, plus free energy. Okay. So now if I go back, I think it's this one. Here you go. All right. We consume ATP. Again, we make ATP from ADP. All right. I know it's confusing right now, but we get ATP. It breaks down to ADP. We take that ADP, make more ATP. But this is what's happening right here. Okay. Is we're cleaving that gamma phosphate, the gamma phosphate, to release the energy that the sun made from fusion. Okay. This energy right here. And that's what ATP is doing. It stores that energy from the sun, breaks it apart with hydrolysis through water, energy is released. And that energy is what allows us, I think I'm on slide 7. No. That energy is what allows us to do, and let me get an ink color here. This energy is how we do muscle contraction. That's terrible. Is how we do muscle contraction. That's how we think. Okay. Um, that's how we do cell transport, the active transport across our cell membranes, how we transport the potassium and the sodium against the gradient. Remember, we talked about that last week. Cell division, mitosis. We'll talk about that in future weeks. All right. This is what's happening here. So remember Hess's law. I break down glucose. Let me go to, where's Hess's law here? Did I have it? Yeah, Hess's law is I start with my energy, my glucose, and I take step-by-step reactions to break it down slowly. This is why we don't break glucose down in its whole. We break it down step by step to make ATP, which is essentially like using pennies to get something as opposed to just throwing $100 bills at something. You're going to burn through it too quickly. Okay? So glucose broken down through many metabolic steps does not produce more total energy. The advantage of this is if I do it step by step by step, I can capture energy gradually as opposed to a fire where I lose that all by heat. Okay, that's where Hess's law comes in. And that's why I think it's important. Again, um, let me change this. So muscle contraction, all this stuff. Let me get to my white. Um, ATP is an efficient way to capture energy, all energy, and not lose it as heat. Okay? Whereas if I just burned it all at once, I'd get too hot. All right? Awesome. We're moving right along. Um, so yeah, so one example is, and let's talk about the active transport, because your book does talk about this. When I pump stuff across the proton gradient, or not the proton gradient, your cell membrane. So you remember I have that sodium potassium pump. All right. Well, the sodium potassium pump, remember, it pumps across the gradient. Remember, sodium is in excess inside or outside the cell. Potassium is in excess inside the cell, but the pump moves it across that gradient. I'm not going to go further into that, but like if I have I'll do another color just to make things a little bit easier to see. So here's my cell membrane, and I'm going to make this sort of rudimentary pump here. Okay. Okay. So you got to remember, um, you have a bunch of sodium that is outside the cell. So sodium outside the cell, and you have, or sorry, I get this backwards all the time. Yeah, you have sodium outside the cell. So the system wants to move sodium this way, and your pump moves potassium this way. Sodium, potassium, like so. Now, This requires ATP to make this happen because remember, you're moving against the electrochemical gradient. You remember there's more sodium outside, so the system doesn't want to move sodium outside, or it doesn't naturally want to happen. So sodium moves against the gradient with ATP. You spend some ATP, and you can move 3 sodium out and 2 potassium in. So that's kind of how that works. You move 2 potassium in, you move 3 sodium out for every ATP that's spent. That little chemical formula, that little chemical ratio is very important. So you should know that. I spend 1 ATP for active transport to move 3 sodium out of my cell and 2 potassium in to maintain that electrochemical gradient. Again, if I don't have that, if I can shut down these proton gradients or these proton pump or these active transport pumps, my cells die. I die. So again, you don't eat, you don't get energy, you can't move this electrochemical gradient, and you shut your cells down. All right, and that is going to bring us to our last part of this talk, where we're going to talk about enzymes and what does this mean when it comes to energy, Gibbs, all that stuff. So, um, a lot of biological reactions are thermodynamically favorable. Um, Glucose, again, disorder, when we're going from order to disorder. So if we're going from, uh, remember, so according to the second law, we favor entropy. So entropy. So if we break down polymers to monomers, this is something that the universe tends to want to go towards, is this idea of disorder. Okay. So if this stuff breaks down, if ATP, again, ATP is highly ordered, ADP is a little more highly unordered. So ATP to ADP plus that inorganic phosphate, all these are energetically favorable. They want to release energy. So if these things don't happen, or if these things aren't happening just all the time, why is that the case? And we've got to go back to something we talked about earlier. It's because we have this thing called activation energy. Okay. Activation energy is sort of like a governor on a golf cart, if you've ever driven a golf cart before, right? So if I'm going to go back to our old example here, if I can take my little hill here, and then I got my ball that's up there. Okay, it has potential energy in the fact that it would like to exist down here, but it has to overcome this barrier. This barrier we call activation energy. So right there is activation energy. Every system has activation energy built into it. And the biological solution to activation energy is the enzyme. Okay? Enzymes, right here. So enzymes can speed up or slow down reactions. And it's all RXN. Sorry, that's reactions. Enzymes can speed up or slow down reactions by lowering or raising that activation energy. Okay? Most enzymes are proteins whose job is to speed up a chemical reaction by lowering that activation energy. Um, without this enzyme, without these enzymes lowering activation energy, biological processes would be much slower, and they would cost more energy, would be more slow. I think that's a better way to put it. Without enzymes, of course, okay? I'll just do without enzymes. So enzymes help lower that activation energy, okay? So without enzymes, it would still be possible, but they would occur far too slowly for life. So what does this mean? So let me take, and let me kind of drift right here. So the idea of enzymes is you have an enzyme and a substrate. Okay? What does this mean? Enzymes bind to certain substrates. Um, the enzyme does the work. The substrate gets worked on. Okay? Enzyme does the work. The substrate gets worked on. Um, an example of this, um, and let me just go to some enzyme examples. Okay, some enzyme examples. Number one, you could have something like lactase. We've talked about this, okay? Lactase is the enzyme, okay? This is the enzyme, and lactose, or dairy, however you want to look at it, but lactose would be the substrate, okay? So our body, in order to break down lactase break down lactose, um, into glucose and galactose, but to break that down needs this enzyme lactase because the activation energy to do so is just too high. So again, we have lactose here, and to get down to glucose, you got to overcome this activation energy. Well, what lowers that activation energy? So what makes it more possible by doing something like this, or we'll just even say getting rid of it altogether. I'll do a different color just to show the difference here. So now you have lactose to glucose, and this was all made possible by lactase lowering that activation energy. Okay, so that's one example. Um, and I could put any enzyme substrate there. Another example that you could have is something like catalase. Catalase is an excellent one. You've seen catalase in action. Okay, so catalase binds to um, hydrogen peroxide, and it makes it into water and oxygen. So imagine whenever you put the hydrogen peroxide on a cut and you see those bubbles happening. Well, that's catalase interacting with hydrogen peroxide. So catalase is the enzyme, hydrogen peroxide is a substrate, and the bubbles, oxygen, water are produced. Okay? There's other types too. So you have, let's see, go three. You have ATP synthase. Okay? So remember, initially we have ATP, and that produces ADP plus inorganic phosphate. However, I have to recycle all that stuff. I have to recycle all that stuff, and I would have to take ADP to make ATP. Okay, because I get energy out of this too, which means I need to put energy back in. Well, ATP synthase helps recycle ADP plus your inorganic phosphate plus your energy to get ATP. Okay? So again, ATP synthase is the enzyme that binds to ADP and inorganic phosphate to make ATP. Okay? So ATP synthase combines those two. So you have enzyme substrate. So going back here, you have enzymes and substrates. Okay? And your enzymes bind to your substrates. Okay? So how do enzymes work? Well, they lower the activation energy. We already talked about that. They lower the activation energy. What they don't do is they don't create energy. So they don't create. Remember, you cannot create energy. They don't create. They don't change the amount of energy. So delta G, they don't change delta G, your free energy. Okay. Um, and they don't change the amount of energy. They don't create or change energy. Okay. They just lower the activation energy. Okay. So if we go back to Hess's law, if you remember that, the pathway doesn't affect the overall energy. So just lowering that substrate or that energy, activation energy, doesn't change the amount of energy that's released. Okay. To put it similarly, we're still going from here to here, regardless if that hump is there. Okay. That's the difference. So here to here, the energy is the same. All right. So enzymes and substrates, they don't change delta G, they don't create energy. So what's happening here? So enzymes and substrates, we're going to section this off so you can kind of see. Um, every single enzyme that you have or that's in your body depends on this thing called the active site. Okay? And the active site is if I were to draw my ghetto little enzyme here, it looks like a Star Wars symbol. Um, this is the enzyme. Okay? And then I have something, then I have a substrate that would bind to it. Let's get this blue. And I'll just try to trace around like so. And this is your substrate, okay? Okay, and that's how that kind of works. An enzyme will bind to a substrate and it'll catalyze a reaction. Remember, catalyze, it'll help break something down, all right? It helps, so it's catabolic process, okay? And then what can end up happening is once that's done, once that's broken it down, again, let's just pretend this is like lactose, the substrate can peel off and then become its individual constituents, okay? So that's sort of what's happening here. You have this active site on the enzyme that the substrate binds to, and it depends on the enzyme, okay? So this is what's happening, and every enzyme is specific to a particular substrate, which is really cool. So the enzyme for this, we could pretend this is lactase, and then these are going to be your glucose monomers under here. Okay, so lactase, converting lactose into glucose. If you don't have, if you're lactose intolerant, you don't have the lactase enzyme, or you have a lot less of it. Okay, so this is actually kind of important. Now, what I want you to remember here is these enzymes. What I'm kind of showing you here is this old model. It was called the lock and key fit, which simply means that these little areas, these active sites don't change. It really doesn't operate like that anymore. Or we know it doesn't operate like that because enzymes are essentially proteins. And what we've noticed is that, let me get a different color here. Let me get this color. And let's just pretend that this enzyme is the exact same here. We're going to go a little different shape. Okay? As the substrate approaches it, let me get the blue. As the substrate approaches it, it doesn't automatically have all the right shapes to it, okay? So as you can see, this doesn't quite fit up, but as it approaches, the system will contort. The hydrogen bonds will shift again because as stuff starts to approach each other, as these molecules in the substrate and the molecules in the enzyme begin to approach each other, you contort, um, and you get oriented differently in space, and then they end up molding together pretty nicely. So just pretend these are now molded together when initially it didn't look like those two would fit together. This is the induced fit model. Okay. Your book talks about this. And it's the same concept. It's just that as they move closer together, the active site then orients itself to accept the substrate. Okay. So that's the difference right there. They change shape, the binding becomes even more precise. And this is ideal conditions for a reaction to occur. So it's more molded, it's more tight. Okay. So Why do enzymes matter? So let's just, well, I mean, we talked about this. So why do enzymes matter? Why do enzymes matter? Okay. Well, they matter again because when we talked about combustion, remember, we talked about combustion, C6H12O6 plus some oxygen is going to yield H2O plus CO2 and energy, right? That is the same model. That is combustion, that is respiration. It is the same model right here, okay? The difference is if I burn fire, I release all that energy out as once. With enzymes, I can control the release of energy. And so I can slowly do it. It's more efficient. I don't lose as much energy to heat because remember, heat energy transfer is very imprecise. You can lose a lot of that energy in the form of heat. So it's controlled combustion. That's the way to look at it. Controlled combustion. Okay. So this is why cells don't run at maximum speed. If enzymes are effective, why doesn't every cell operate at maximum speed? Because cells need control. We regulate these enzymes too. We can't have enzymes running around everywhere. So how do we do enzymatic control? Well, your body has some, or your cells and the metabolism your body runs through, very cool. So you have a few things. You have one thing called competitive inhibition. You have non-competitive inhibition. And finally, you have something called allosteric or feedback inhibition. Okay. And feedback inhibition. Okay. And going back, section this off so we don't get confused here. Going back to our site. So we're going back to our enzyme here. Now I kind of drew a rudimentary enzyme here. Okay. Here's our enzyme. And ideally, we would want to see a substrate that fits in beautifully. So if I go just kind of drawing that through like so, this would be a beautiful substrate. But if we could give a drug to somebody that has that exact same shape, this could also be an inhibitor. What do I mean by that? If I give somebody a drug that binds to this enzyme, it'll prevent the substrate from binding to it. Okay? An example of this, so with competitive inhibition, an example of this is statins. All right? With statins, um, basically what you end up getting is your body can't create cholesterol. We give them statins, and that inhibitor binds to the active site of this enzyme. Okay? And the substrate can't bind to it to create cholesterol. So their cholesterol is lower. This is a statin drug. Competitive inhibition, because the inhibitor competes with the substrate for this spot right here. Okay? So that's a competitive inhibition right there. The other type you might have um, is something called a non-competitive um, inhibition. So what ends up happening in non-competitive inhibition, let me get my yellow here, is let's pretend we have like these two sites. Let's just pretend we're making a Batman signal here, okay? Now, here's your active site here, active site, and you have this other site on this side over here. The idea behind non-competitive inhibition is that I have a drug or a poison or a toxin that will bind to this site like so, and then change the orientation of this side up, the active side up, so that the substrate can no longer bind to that. So no substrate can fit to this, or the substrate that was intended to fit to that no longer binds because I've oriented the protein differently, and this site no longer binds. No binding. So this is non-competitive. This is non-competitive because they don't compete for the active site. An example of this would be like cyanide. Okay, cyanide blocks the electron transport pump and prevents your cell from generating energy, generates that ATP, that proton gradient. It prevents it from happening, and you don't get ATP. Your body shuts down. Okay, heavy metal poisons are the exact same way. Mercury, lead, they interfere with enzymes by altering their shape. Um, and then you have something, and if I can, do I have room here? Yeah, I have room. I'll make room. You have something called, let me get my whiteboard. So we'll have something here called allosteric, which is essentially like a dimmer switch. So these are like volume controls. It's separate from the active site. So like so again, here's our substrate, but then you might have this other allosteric site over here. And this allosteric site is like a dimmer switch. This is what you can speed up. Again, if I get something to bind there, it can speed up or slow down a reaction. So this is allosteric site or allosteric inhibition or promotion allows for enzyme activity to go up and down depending on what is bound there. Okay, so you have competitive inhibition, non-competitive, allosteric sites or allosteric inhibition. Um, and so what a good example of this is would be, and again, I'll just do this, is ATP and ADP. Okay. So really, there's an enzyme, this ATP synthase. We'll just call this ATP synthase. Okay. ATP synthase converts ADP to ATP. Okay. Hopefully that's not good. Hopefully I'm getting some. All right. Converts ATP to ADP. That's ATP synthase. Okay. When ADP is up, so this is an allosteric inhibitor, ADP will bind to that site right there. It will take ATP synthase to stop. If there is lower ADP, that means that nothing's bound there, and ATP synthesis goes up. So the presence of more ADP, as I use more energy, and ADP, I think I got that backwards. As ADP binds to it, there's more ADP. That tells the enzyme to make more ATP. If I have an abundance of ATP, that means I have less ADP, and then production of ATP goes down. Okay? So ADP is an allosteric activator. It promotes ATP. ATP is an allosteric inhibitor. It tells ATP synthase to stop. Okay? And then you have something kind of similar, which is just that feedback inhibition. And running out of space here, so I'm going to section this off right here. And you have feedback inhibition right here. And that just basically means as the more I get something, it tells the enzyme to stop. So more stuff, more product, less enzyme activity. So more product, less enzyme activity. There are a few examples of this. Isoleucine is a big example. Cells use enzyme to produce isoleucine. So here, and that's just one of your amino acids you'll learn. Isoleucine. Isoleucine. Okay. As isoleucine goes up, the enzyme production goes down. So the product is the inhibitor. And as isoleucine goes down, the enzyme activity goes up. Okay. So that's just the final summary of enzymes and regulation. Okay, is enzymes are proteins that bind to a substrate like lactose, ADP, whatever. They promote or lower an activation energy, and that allows the uh, the the reaction to take place. Okay, so uh, keep that in mind. Okay, so again, you have competitive bind to the active site. Two things that combine, they're competing for the active site. Non-competitive binds to another side of the active site or another side of the enzyme, contorting the active site, which prevents the substrate from binding. You have a dimmer switch of sorts, like an allosteric inhibition and activation. ADP, ATP is an example of that with ATP synthase, and feedback inhibition, where I get more of a certain product that binds and allows the enzyme to be more active. So feedback inhibition, all those are very important. I should be able to say those in a question, in a sentence, and you answer it or you know what I'm talking about. All right. That being said, that concludes our talk today. So if you have any questions, please do not hesitate to reach out to me, and I will be happy to answer and hopefully clear up some of the muddy waters I may have made. But as always, this is me signing off, and like I always say, stay curious.